So our first speaker is Matreyek. I think we're all very excited to have him. He was recently here as a postdoc in Doug Fowler's lab. He got his bachelor's at UCLA and then did his PhD in virology at Harvard. And while he was here, he had a huge impact, at least within the genome sciences department in the deep mutational scanning field. He developed a mammalian landing pad system, which allows us to integrate single genetic variants into mammalian cells, which has had a huge impact on DMS experiments. And then he also developed VAMP-seq, which is a DMS technology to assess protein stability of variants. And we're really excited to have him back, and we're excited to hear what he has to talk about today.
Great, thank you, Flo, for that introduction. So yeah, as Flo said, I'm Kenny, and I'm in the currently in the Department of Pathology at Case Western Reserve University School of Medicine. And I just started there 4 months ago, and before that, I was in Doug Fowler's lab. And yesterday we heard about all the amazing results that are possible by people performing the different types of mutational scans. And while it's a powerful approach that's going to give you a lot of data, a lot of insight, there is certainly this very technical aspect to it, which I think will be the major emphasis of today. Part of my goal for my talk today is to think about or bring up some of the kind of strategies and kind of best practices such that people that want to do this approach can do so with the least amount of cost, pain, and suffering. So if you're here, then you're already likely pretty familiar with deep mutational scanning, but I still like to go over some of the basics at the outset. So traditional approaches are typically confined to a tube, well, or vessel that maintains that link between genotype and phenotype. And if you want to study a variant, compare it to wild type, then you have to make it in 2 separate tubes, and you have to make sure that you label the tubes appropriately. And if you forgot to label them or switch them up, then you're essentially out of luck. And while it's great for a handful of samples, it doesn't scale particularly well. And testing more samples generally entails taking an arrayed approach into smaller and smaller vessels to conserve resources. So you can think of 96 well plates or 384 well plates. You need to keep that plate map current to interpret your results. If you want to go more than that and test hundreds of thousands of variants, then essentially you need automation to be able to do it because your hands would essentially fall off. And not everyone has access to these machines, and these things become pretty expensive as you try to scale up to that level. And part of that beauty of deep mutational scanning is you're now able to get away from these individual tubes or wells and work on units that inherently scale much better. Since you can take a pooled format that tests all the samples within a single plate or well, for example. So instead of genotype-phenotype links being confined to essentially the vessel, deep mutational scans can exploit links at much smaller scales. So in the case of scans in bacteria, yeast, or mammalian cells, this requires turning the experiments essentially into a single cell format where an individual cell encodes a single variant and also exhibits the properties of that variant. And that's the context I'm used to thinking about. So I will admit that most of the things I'll talk about today will be slightly biased along those lines, but yeah, so I apologize to some of the people working other scales. But there are other scales that are certainly doable. So you can imagine single molecule or single protein complex assays, such in the case of ribosome display, where the mRNA is encoding your genotype, and then the peptide or protein that's on the stalled ribosome is encoding the function that you care about. And also, there are also other approaches which I think are still a little bit more in their infancy, but I think will be adapted further on as we do more mutational scans, such as droplets, which are able to potentially encapsulate kind of soluble phenotypes, which we haven't necessarily been able to do with these single cell approaches. Notably, when performed on the cellular level, both DMS and CRISPR screens are single cell assays, so functional assays that work in one could potentially work in the other. And I think that's a nice thing to keep in mind where, if you fully vetted assay for, some biology you're studying in one aspect, then, consider trying to think about, well, would it be interesting to do a genetic screen in that as well? So deep mutational scans share a few key steps, although there are now enough derivatives of this that there are essentially exceptions at most of these steps. So first is creating your library of variants. Next is expressing that library of variants within your model system, whatever that may be. Could be many different things. Third is separating the variants based on a property of phenotype that you're interested in, that's captured in your assay. Fourth is using high throughput sequencing to count how each variant has separated based on that assay or protein of interest. And lastly is the big one, which is analyzing results to actually gain insights into the problems at hand. And there are many considerations that need to go into each of these steps of a demutational scan. For example, what library do you want to test as a site saturation mutagenesis type of library where you're testing every possible amino acid, or do you mostly just care about ones that are possible through single nucleotide variation because you care about variants that you might see in people? Do you only want to look at single variants, or do you want to understand double variants, for example, look at epistasis? Also keep in mind, these aren't mutually exclusive. I think you should kind of hedge your bets to take the library design that is going to give you the best quality library for what you're interested in, but you can certainly have a library that has both, in which case you might be able to essentially reuse that data set for a couple of different studies. Do you want to barcode your library, which is a little bit more cost up front and less cost later on, or do you want to just do direct sequencing? And are you worried about getting the best results possible and having the most comprehensive library and nothing else will suffice? Or are you good enough, or is it okay to have good enough results if it's vastly cheaper? There's also the question of model systems. So what's the best model system to study your problem? And again, I think you don't necessarily have to have just one. If you think that there are potentially multiple model systems you can go into, you can design your library such that it'll be easy to shuttle it into a yeast vector as well as, say, a mammalian vector. What assays can you use, and how can you vet it early on such that you can be very confident that will work in a deep mutational scanning type format? And at the end of the day, what do you hope to get out of your deep mutational scan? And I think I'm going to focus on that first, since that's by far the most important question and essentially determines everything else in your deep mutational scan. So if you simply care about protein binding and don't want to extend analyses beyond that, then you can essentially directly test for that phenotype in your assay. And a lot of model systems that will make natively folded protein will work for that. On the other hand, if you care about things like functional interpretation of clinically observed variants in people, then extra care must be taken. So you obviously can't perform scans on humans. So the next best option is to find an experimentally tractable, physiologically relevant model that's capable of correlating with disease severities and outcomes observed in patients. And while many models can achieve this, if done correctly, I'm particularly excited about saturation genome editing, spearheaded by Greg Finley and Jason Duri's lab, as a great example of this, because it captures a lot of the aspects that you'll get in genomes with variants, such as mutations of splice junctions, intronic variants that might matter, or nonsense mediated decay. But regardless of what you choose, you'll certainly need good clinical data for validating the utility of your functional results. And there are now a number of useful resources to help guide you with this, including this paper that was published recently in Genome Medicine. And I also suspect Brian Schertz's presentation later on today will certainly touch on this and give a good perspective. So there will always be trade-offs, with each model system having its own advantages and disadvantages. And typically models with more complex organisms, which are larger, harder to manipulate, will handle somewhat smaller libraries. But I also think that familiarity with your model system is also a very important factor, since there will always be a lot of troubleshooting with the experiments that you're doing, and being able to really understand how your system could be going wrong is a very valuable asset. And Ethan Ehler, who's previously done work in both yeast and human cells, will talk about this in more detail a bit before lunch, so I won't go into much more detail here. Another major consideration is how important it is to have good quantitative scores for all the members of your library, versus tailoring your experiments to just find a handful of the most interesting variants. So if you need, you need comprehensive scoring to be able to create these complex sequence function heat maps that we all love to look at, and to perform more advanced analyses that need good coverage, where missing data is not going to be compatible. But getting good coverage libraries and results takes a fair amount of focus and dedication, and surely isn't the most efficient way if what you want to do is explore a vast amount of complex sequence space. And other times, deep mutational scans are more of a means to an end, and comprehensive maps beyond the scope of the experiment are beyond the scope of the experiment when all you want to do is find an improvement. So accepting this fact early on will allow you to test larger libraries since it's much faster to find a needle in a haystack by floating a magnet over it and seeing the things that are able to stick rather than actually having to pick up each individual thing and figure out is it a needle or is it a stick of hay. And put another way, the human immune system doesn't care about making sequence function maps when it's trying to do somatic hypermutation for affinity maturation of its antibodies. It just cares about keeping you alive. Different subtypes of DMS-compatible assays have their advantages and disadvantages. So growth and survival assays are relatively cheap since they typically just involve making replicate plates that you want to, for different conditions you want to test or different time points. And they can also handle gradations of effects nicely since that information is not inherently lost in the actual readouts. But not all growth assays are created equal, and they can be divided into, say, like positive and negative selections that either enrich for a property of interest or where your property of interest is depleting within your pooled sample. And since the results are read out by sequencing, negative selections are by far more costly since you have to sequence far deeper to watch something deplete in a sea of other things they don't care about. And it's also just kind of statistically harder because, yeah, I mean, it's harder to better quantitate that type of effect. And this can be kind of complex and tricky, and it's worth going over some potential outcomes to really figure it out. So I just quickly try to come up with some examples. So here's one where, say in HEC293T cells, you overexpress a tumor suppressor like RB. So if I do that, then cells that express wild-type RB do not divide, and they essentially croak, and they fall out of the pool. Versus loss of function variants that don't have this effect, presumably they'll continue to divide. But of course, things aren't that simple, and there will be some cells that just didn't get transformed or recombined, and these will continue to grow and also kind of confound that pool of cells that you have. Libraries also have numbers of indels and frame shifts. Again, in this case, they will continue to divide. And also, not everything expresses perfectly. There will be a distribution of expression, and some will just have less wild-type RB expression, or will have silenced their RB genes, in which case they will also divide as well. So, I mean, this format could maybe work as long as all these confounding factors end up not being an issue because your library is really high quality. But if they are in there, that they could certainly make it a lot harder to get meaningful results. So in contrast, say there's a different hypothetical situation where, say it's a cell survival protein that you're mainly interested in only the active variants. So only these variants in cells will allow the cells to survive and thus enrich, in which case all of these other circumstances will then be depleted. So I think this will probably be a format which would be more robust and likely be easier to get some good results out of. An arguably more flexible selection is to use machine-based sorting like FACS or fluorescence-activated cell sorting. And there's a lot of single-cell fluorescence-based assays that are already out there and well-vetted to study different types of biology, and these can be easily integrated into deep mutational scanning type experiments. Furthermore, it's an immediate readout, and it's easy to track and interpret. You can see if your cells are green or not right away instead of thinking that they might be depleting later on when you're actually reading it out. And while most universities and research institutes have decent FACS capabilities, it can be rather expensive to spend hours in front of a machine and also kind of soul destroying and limitations to the number of cells in that format can make it a bit harder to get all the gradations, as well as if you're trying to do, say, four-way sorting to try to get the gradations, you're limited to the number of gradations, and thus it could make it a bit harder to get really good, accurate quantitation across the board. So the assay you choose will be highly dependent on biology that you're trying to study, so it's hard to speak specifically here. But it's always safest to go with well-vetted assays described in the literature, and ones already used in a single-cell format are perhaps the most likely to work. And next are those that have been used in a traditional or bulk assay, but by numerous groups, but are still theoretically compatible with, say, a single cell format if you're working with a cell-based model. But there will always still need to be careful preliminary experiments showing that it will work in this pooled assay. And essentially trying to do a mini experiment of your DMS before you scale up to a full library, I think is a good idea to get a sense of, will this actually work in the full-fledged experiment. You can certainly build your own assay, but keep in mind the assay development itself can be a slog and a struggle, and in many ways can be considered its own advancement independent from the mutational scan. And lastly, this is perhaps the point more relevant for the analysis step, but always know and communicate the limitations of the assay that's being used. So in the case of these VAMP-seq experiments that I did in Doug's lab, we tried really hard to communicate to people that abundant variants could still be non-functional and be pathogenic when observed in people. And instead, the true value of it was the opposite situation, where the loss of abundance variants we thought were very likely to be non-functional and thus potentially pathogenic. And I also think there's a lot to gain with multiple assays on the same library. So an example of this happened to me back in 2018, where Taylor Miguel from Brian O'Rourke's lab published a DMS on P10 phosphatase activity in yeast, and I published a different scan of P10 intracellular abundance in hex cells. And we focused on different things, and these were largely complementary. But I think there was some value in being able to compare these two data sets. So some of the inconsistencies between the two assays were less reflective of biology, at least in my opinion, and more about the details of the assay. For example, I think a large number of the unstable and active variants, which are colored in blue in that top left, were likely just reflections of the dynamic ranges of the two assays, where the abundance assay was best at quantitating destabilization from the wild-type sequence, whereas the activity assay was more quantitative when trying to look at even just any activity-based phosphatase activity rather than a dead enzyme. So even very low activity, I think, was still scoring in their assay. And this won't always be the case. So people with sharp eyes yesterday may have noticed that the relationship was seemed to be opposite with cytochrome P450 data that Clara presented, where there were many partially abundant proteins that had little activity in their functional tick assay. On the other hand, different activities in the two assays can have biological significance as well, and can help you better discriminate different types of variants from one another. So for example, I could focus on this subset of stable but inactive variants on the bottom right over there, colored in red, since those are potential dominant negatives of P10. And this is interesting in P10 because they could potentially be, or it's been thought that dominant negatives could actually have a more exacerbated effect in various diseases. So in some unpublished data that I created right when I left Doug's lab, I think I even did the Western blotting after I'd already left. I took a number of these variants and expressed them within HEC293 T cells where there was already a wild type copy, and then see if saw if any of them had potential dominant negative activity. So the ones on the right over there are the previously uncharacterized ones, and it looks like a couple of them perhaps do have dominant negative activity, resulting in increased phospho-AKT in the downstream pathway. So I think having multiple data sets, I think, can be a very powerful approach to better discriminate different types of variants from each other. Over the years, I've gotten many questions about avoiding bottlenecks in the workflow. In many cases, how many times you want a barcode or how many times you have to recombine cells. And people generally want general rules such as 100-fold coverage of variant library. And I think while that's convenient, that it kind of gets away from the actual how you should really be thinking about it, which is that I think you can just consider the various steps in DMS as various sampling steps. For example, over here, I'm taking 1,000 variants in this uniform distribution and sampling 100,000 events, and that's the type of distribution I would get if I did that. But uniform distributions are hardly realistic, and if you instead sampled 100,000 events from a distribution which is that you get from real life libraries such as the log normal distribution of my P10 library, then essentially the results end up looking like this, where with 100,000 events you're still seeing essentially all the variants, except on average a lot less, and there are definitely a number of variants that are there and vast numbers, which could potentially kind of take over your experiment. And if you don't have this information yet, I mean, I would say that, at the library QC step, you should sequence your library and figure out the distribution and how many, what your coverage is of your variants, as well as how many wall types or indels, but also see, what's the distribution of your missense variants. And if you don't have that data yet, well, you can, at this point, there's enough people that have published this kind of data that you can probably use their libraries, as long as it's similar to yours, as a proxy. And in my case, I'm comparing two different libraries. So again, the P10 one, and this is the frequency distribution in black. And then I'm also including another library I looked at closely, which is the NUD-T15 one, which was published currently on bioRxiv by Chase Suter, who, when he was at St. Jude, but now he's in the MCB program here, where they had a much tighter distribution, probably because they bought their library from Twist. And I think, you can use these types of distribution for your ballpark calculations. And you can look and you can see that if I then try to sample, say, I wanted every variant to be present in a cell at least 10 times. Well, you can, try different numbers of recombined cells and see, well, how many recombined cells do you need to actually get to pretty good coverage of your library. All right, just to finish up, I wanted to say a few things, which is that, I think science is best when it's open and reproducible, and I think this community has been good about that in the past, and I hope it continues to be that way in the future. I think some general best practices are, of course, publish your primary data on SRA or Geo. Raw data is great, but many people trying to actually analyze your data set will rather start with the processed data, so of course have that as well. You can include that as a supplementary data file with your publication, that's good. And also Geo has a spot for this, but there are also additional user-friendly ways to present this, such as your lab website, which we've done numerous times, and now also MaveDB, which I think will be a good repository for this. And also, this type of analysis inevitably requires a fair amount of, or this type of studies requires a fair amount of analysis pipeline, and it's good to share that as well. So I think, again, supplementary data files could be useful, though I've noticed that many journals are very clunky with this. So having it up on GitHub, I think, is great. And also using established software can be a nice way of having it be reproducible and having everybody be able to kind of go through those steps again. Where Rich2 is a good example of this, developed by Alan Rubin. Every time I've done a deep mutational scan, I've always run my stuff through Enrich. So these topics will be covered in more detail by people coming after me. So if there's more specific questions about those, you might want to wait until then. And I think it'll be exciting to see some of the new techniques that are now available as well, such as dropsynth, which we heard a little about yesterday, as well as visual cell sorting, which Nick's been spearheading. And I'll stop here and take any questions, although I also want to put in a shameless plug, which is that if you know anyone that's interested in cell engineering or deep mutational scans, especially in the kind of immune protein sphere, and are willing to live in the Midwest, then I've just about finished equipping my lab, and I'm looking for passionate trainees or scientists at multiple levels to work with me. So I think, oh, didn't mean to actually go there. I'll just leave that up, and then if there's one or two questions, happy to take.